Hello everyone. As you have completed with the important two DNA viruses, that are the hepatitis virus and the herpes virus, we shall now move on to the other miscellaneous DNA viruses. The viruses that we would be covering today are Parvovirus, papillomavirus, Adenovirus and smallpox virus. We shall start with Parvoviruses. Now, parvoviruses, they are the simplest animal viruses infecting the humans. They are the smallest viruses whose size ranges from 18 to 26 nanometer. They are non-enveloped. They are single-stranded DNA viruses whose symmetry is icosahedral symmetry. Now, among all the human parvoviruses, the most pathogenic human parvovirus is the Parvovirus B19. And we shall cover the pathogenesis and the manifestations of the same. Now, if we see the clinical features of Parvovirus B19, we can see that in the initial phase, the features are fever, chills, headache, myalgia, which is then followed by clinical features like rash and arthralgia. Now, this Parvovirus B19 produces certain hematological changes because it infects the precursors of the red blood cells, which then directly results into red cell destruction and inhibition of erythropoiesis, which is profound in the presence of the underlying immunosuppression and hemolytic anemia. If we see for the diagnosis of this virus, molecular is the best option, that is the PCR, as well as ELISA is available for antibody detection. As you can see here, IgM antibody appears early, which indicates recent infection and remains elevated for about 2 to 3 months, while IgG antibody appears late and it remains elevated for a longer time. Now, as I mentioned, the typical clinical feature, that is the rash that appears in the later stage, you can see here the rash on the face as well as lace-like rash on the extremities. Here are few more examples of the slapped cheek appearance. It appears as if someone has slapped on the cheek. The other important virus is the human papillomavirus, whose size varies between 50 to 55 nanometer. It is non-enveloped. It is a double-stranded DNA virus which is icosahedral in symmetry, and this virus has got a selective tropism for the epithelial cells of skin and mucous membrane. Now, these warts, which are basically the benign lesions of the human papilloma viruses, they are classified by two ways, based on their location on the body as well as based on the appearance of how they look. So, here are some few examples. The common wart, which is located on hand and knuckles, the plantar warts on soles or toes of the feet, the flat warts usually on the leg, hand and face, the filiform wart usually on the face near the eyelids or lips, the mosaic wart, the periungual wart, the oral wart within the oral cavity, and the very common genital wart, which is located near or in the genital areas. You can see the example of common wart. the flat warts which are found on face, neck, arms, back of hands and legs. You can see the growth is flattened. The plantar warts, as the name suggests, on the soles, and the genital warts which are found on the shaft of penis, vagina, vulva, cervix and around the anus. There are more than 100 types of human papillomavirus which are recognized based on their DNA sequences. Out of these, more than these 100 types of HPV, 30 to 40 are the genital types, and amongst which types 16 and type 18 human papillomavirus are known to cause squamous cell carcinoma of the cervix. They are known to cause 70% of the cervical cancer cases. And type 6 and type 11 are known to cause the benign lesions, as I earlier told you, the genital warts. So type 16 and type 18 are most commonly responsible for the malignant conditions, and type 6 and 11 are responsible for the benign condition. HPV is a common sexually transmitted viral infection, and remember that if the patient is asymptomatic and if the patient is untreated, it is the main cause of causing cervical cancer. We shall see the pathogenesis in brief. The virus in the cervix enters the cell through the microabrasions. Then several weeks later, the infection spreads. The HPV replicates and the infection spreads to the other cells. While in 90% of the cases, the healing occurs within 2 years, and in 0.8% of the cases, after 10 to 30 years, these HPV invades into deeper layer of the tissues and turns cancerous, leading to cervical cancer. You can see the cervical cancer staging here, the normal front view. The early stage 1B, the cancerous tissue is here. The late stage 1B, it moves further, and stage 2B, the cancer spreads outside the cervix. Now the picture that you can see here is of tree man disease, also called as epidermodysplasia verruciformis. It is a very rare autosomal recessive benign condition, basically a squamous cell carcinoma of the sun exposed areas. As I told you, the benign warts lesion, the warts here, they grow as bark of the tree, and hence the name tree man disease. Pathogenesis of adenovirus. Now this virus, it infects and replicates in the epithelial cells of the respiratory system, the gastrointestinal system, the urinary tract system, the eye and the liver. The spread is via the regional lymph nodes. Now upon infection with the adenovirus, one of the 3 different interactions with the cells may occur. The first is the lytic infection, which eventually results in cytolysis of the mucoepithelial cells. The second is the latent infection, which involves the lymphoid cells or the infection of the lymphoid tissue. And the third is the transformation, that is the oncogenic transformation that has been observed in animal cell culture. Once a person is affected with adenovirus, the immunity that the person develops is quite effective and long lasting. These are the clinical manifestation of adenoviruses in different age groups. As you can see here, acute febrile pharyngitis in infants and young children, acute respiratory disease in military recruits, pneumonia again in infants and young children, gastroenteritis in infants and young children, pharyngoconjunctival fever, which is also called as swimming pool conjunctivitis, which is seen in children and adults, epidemic keratoconjunctivitis, which is also called as shipyard eye, which is seen in adults, and in case of immunocompromised patients or any transplant recipients, the clinical manifestations are pneumonia, hepatitis, nephritis, colitis, encephalitis. You can see the picture of pink eye or the conjunctivitis. The adenovirus is now being utilized in the gene therapy as the gene delivery vehicles. If you remember the basic process of the gene therapy, we need a vehicle that could release the gene inside the cell. And adenovirus is being used as the same delivery vehicle. It is used in oncolytic therapy, gene therapy, and genetic immunization. Moving on to a very important virus, that is the pox virus. If you could recognize this celebrity, the late Shri Om Puriji, he got infected with smallpox virus in his childhood. He then got recovered with it, but unfortunately the pox marks were always with him. Pox viruses are one of the largest and the most complex of the viruses. The infection through these viruses causes febrile rash. There are few important members of the pox viruses, that includes variola, vaccinia, and molluscum contagiosum virus, where variola is the causative agent of the disease smallpox. Now smallpox, as I guess you are all aware, was the very first infectious disease to be eradicated from the world. The WHO declared smallpox eradication in the year 1980. But we still study pox viruses. Why? Because pox virus is one of the biological weapon. After the eradication, as vaccination was stopped, the people who were born after the year 1980 are not immunized, and hence smallpox virus can be a potential agent of bioterrorism. Of course, there are other reasons why we should study pox viruses, but yes, this is one of the most important reasons that we still need to know about the pox viruses or the smallpox. You can see here the complex symmetry of the pox viruses. Their structure neither fits into the icosahedral nor into the helical symmetry. It is basically brick shape or ellipsoid shape. Now if we see the pathogenesis and the manifestation of pox viruses, the infection is transmitted via droplet or aerosol. The incubation period is 7 to 17 days. In the first few 3 to 4 days, the complaint is of fever, which is then followed by ananthem. Ananthem is rash inside the body. Ananthem for 2 days, and it is then followed by exanthem. Exanthem is rash outside the body. This exanthem lasts for 12 to 32 days. Now this pattern of fever, the appearance of the rash, these things helps in differentiating between smallpox and chickenpox. So how is smallpox differentiated from chickenpox? In smallpox, the fever is 3 days before the rash, and in chickenpox, the fever is the same time as the rash. And based on the appearance of the rash, in smallpox, the rash crop at the same stage, and in chickenpox, the rash crop in stages. The rash develops slowly in smallpox and fast in chickenpox. The distribution of the rash is centrifugal in case of smallpox, that is, it is seen on the face and extremities, while in case of chickenpox, the distribution is even. The palms and soles are usually included in smallpox, and it is uncommon in case of chickenpox. The death is 10 to 30% in smallpox, that is by variola major, and it is very rare in chickenpox. You can see the distribution of both. In case of smallpox, the face and the extremities are involved more. that is the centrifugal distribution. Now, as I told you that the lesions of the smallpox, they crop at the same stage of the development. These stages of the development are macules, papules, vesicles and crusted lesion. Here is the picture of an early pustular lesion of smallpox. Variolation was the first attempt of providing artificial immunity against the smallpox. In this process, skin scraping of a smallpox patient was taken and then inoculated into healthy individual. This practice was done for at least 1000 years in China as well as in India, but obviously it was risky. These are the pictures of variolation where you can see the healthy individuals are being inoculated with skin scraping material from the smallpox patients. For many centuries, smallpox has been a very devastating disease, but now in modern times we do not have to worry about that because of this remarkable person, Edward Jenner, whom you all know is the person who discovered the first vaccine. Now, Edward Jenner observed that in milkmaids who were previously infected with cowpox virus, they did not have any signs or symptoms of smallpox, and hence he concluded that using the cowpox we can prevent the illness from smallpox. You can see the very famous picture of Edward Jenner inoculating a 13-year-old boy, James, with cowpox. Smallpox vaccination was done using a needle with a fork. A drop of vaccine is held in the fork of the needle, and then the needle is held perpendicular to the arm and then punctured. After the cowpox vaccine, live vaccinia vaccine came into the use, and it was highly effective. It was given in a single dose between 1 and 2 years of the age. With the use of this highly effective live vaccinia vaccine and by using the multiple puncture technique with a bifurcated needle, eventually in the year 1980 the smallpox was then eradicated from the entire world. And the last one, Molluscum contagiosum, which is again one of the member of the Poxviruses. It causes skin lesions which are pink, pearly, wart-like lesions on the skin with a characteristic dimple at the center. For the laboratory diagnosis of this Molluscum contagiosum, there are typical Molluscum bodies which are intracytoplasmic eosinophilic inclusions seen in the skin scrapings when stained with histopathological stains. In the end, I would just like to mention a rhyme, a nursery rhyme, that is, "Where are you going to, my pretty maid?" It is assumed that this rhyme is a conversation between Edward Jenner and the milkmaid, from whom he thought that cowpox could be a solution to the smallpox. Smallpox is basically a very historical and ancient disease. If you are ever interested in the history of the same, please go through it. You would really like the historical part or the historical aspect of this disease. Thank you.